about my project on behalf of my team, and our project is about cancer therapeutics. We are dealing against cancer. So, what is the problem? The problem is the ovarian cancer. Ovarian cancer is the type of cancer in which number of abnormal cells grow in the ovary. And there are two types of ovarian cancer, type 1 and type 2. Type 1 is low-grade serous carcinoma and is easily manageable with the chemotherapy, while type 2 is a high-grade serous carcinoma and it's not manageable with the chemotherapy. It's highly aggressive and overall survival rate is less than 30%, and worldwide incidence is about 300,000 per year. So we are dealing with the high-grade serous ovarian cancer. As I already told that this is unmanageable with the chemotherapy. So the available treatment for the high-grade serous ovarian cancer is the first-line therapy, which is chemotherapy based on cisplatin, carboplatin, doxorubicin, different type of drugs. But the issue is that this is very toxic. They have many side effects. It's toxic for the normal cells, normal organ. Also, 50% of the patients resist with the chemotherapy, and 50% of the patient relapse tumor after some time of chemotherapy. So after first line of therapy in the market, there is a maintenance with the PARP inhibitor. Those patients who go through first line therapy, they go under the PARP inhibitor maintenance to maintain the therapy, but also it's very costly and they have many side effects. And in the market is a second line therapy. And second line therapy is the combination of the different type of drugs which use in the first line of therapy. For example, cisplatin plus carboplatin and different type of combination of drug. This is also very toxic because if first line of therapy have many side effects, so in the second line of therapy, the side effect become double. So our technology is lie in between PARP inhibitor maintenance and second line of therapy. Our technology aim is to treat those patients who suffer because of the first line of therapy due to the side effect, due to the resistance issue, and also because of the relapse issue. So what about our technology? Our technology is based on nanomedicine. Our technology name is copper nitroprusside, is a medicine. And our material, this type of material, nanomedicine, is highly specific and it's reduced the toxicity. It's only specifically kill the cancer cells, due to which it's reduced the toxicity, which overcome the chemotherapy issue. Also, it's really highly potent and is also destroy the dense matrix barrier. Dense matrix barrier usually is a barrier surrounding the tumor tissue, surrounding the tumor cells, which act as a barrier against the medicine. Usually when we treat any patient, drugs not able to penetrate inside the tumor because of this dense barrier matrix has a sheet. So our technology also destroyed that dense barrier, and our patent is in pending. We already submit and we are just waiting for the acceptance. So how our technology work? Our technology have this structure. This is the final structure of our technology. Our product have together copper, iron, and NO. So how it work and how it's specific? Usually NO is a nitric oxide. It's destroy the dense barrier, which is very important thing. First is our product destroy the dense barrier, and after the destruction of the dense barrier inside the tumor, copper and iron release inside the tumor. When copper and iron go inside the tumor, because in the tumor there is a tumor acidic environment which is acidic, so copper and iron dissociate, disintegrate, and it's breakdown. And after breaking down of copper and iron, it produces copper peroxide. This is chemistry, and copper peroxide produce multiple type of ROS species, due to which we specifically only kill the cancer cell, not normal cells, because the tumor microenvironment is acidic, and our product only work in the tumor microenvironment, not in normal environment. So here's the preliminary data. This is in vivo data we put, and here we see that our material effectively reduce the tumor burden compared to the negative control. This green is our material, and compared to the negative control, while little bit body weight is reduced because our material is bit toxic. And now we prove that this is specifically kill the cancer cell. We cope up with the chemotherapy issue. It's biocompatible, is highly potent, and also is destroy the dense matrix. Overall in the market, overall ovarian cancer cases is about 300,000, and from which 800,000 cases reported die. because of the ovarian cancer. The addressable market is about 70%, because in ovarian cancer, 70% patients are with the high grade serous ovarian cancer, and we are dealing with this case. So this is our addressable market. In the market in America, in Europe, there is about 50,000 and 66,000 cases reported per year with this cancer. And our market entry is about this 70%, because from these patients, 70% and 50% in Europe relapse and resist with the first line of therapy. So we will enter in the market to target those patients who suffer with the first line of therapy. In the roadmap and financial analysis, we are now in the preclinical trial. We already did our experiment in the mice model, and now we are forward to product our scale product to scale up our product and to produce more biocompatible. Then we will go in the clinical trial in phase 1, phase 2, and phase 3. As this is breakthrough technology, we need also ISO approval to enter into the market. This is our team. Our team composed of 5 members. Me and Mohammad Adil are the postdoctoral researcher. Flavia is our mentor. Isabella is a senior researcher, and one person is from the TTO staff. And overall, in our team, we have cumulative experience of 20 years in oncology and nanomedicine. In the wrapper, we can say our breakthrough technology selectively kill the cancer cell, is very highly specific, minimize the side effect, and also is destroy the dense matrix. In the end, I can say cancer is a word, not a sentence. Thank you.